Hmm? Okay, is that better? Okay. Did people find the readings on Slack already? Okay. You can use Slack to ask questions of the instructors or each other, start discussion topics, whatever you want. So to use it, I will get started. Okay, so what I'm going to do now is going to be a lot of basic stuff. It will be some repetition of what Kavita has already discussed and brought up. But the goal here is to make sure everybody's on the same page, that if there are problems that you have or any confusion with a lot of basic terminology, this is the time to get it sorted out so that you can understand the future lectures much better. Okay, so please feel free to interrupt and comment. Good, so some genetics. A bunch of you are not biologists, few of you are not biologists. So again, stop and pause me wherever things are not clear. What is genetic variation? Anybody want to quickly say what it is? What do we mean when we say, oh, this population has more diversity, more variation than others? Come on. I think Kavita's already threatened you with no lunch, so. More polymorphic sites, that's one way, okay. What is polymorphism? Sorry? Difference in the sequence. Okay, good. So I think most of you have this sort of visual in your mind already, but in case you don't, our DNA is like a long string, right? There are bases which form this long string, A's, T's, G's, and C's. And in RNA, this becomes A, U, G, and C's. And this string is really what we're talking about when we're thinking of a genome, right? So picture a long string, which is very, very condensed inside your cells. That's your genome. And when we're talking about genetic variation, we're talking about differences at the sequence level, at the base level, in these pieces of DNA which all of us have, right? By and large, how much variation is there in this room? Can you tell me how much variability is there in this room amongst people here? Roughly? How many mutations probably separate each of us? Any pairwise combination? 70? Less than 1%. Why 1%? Where is this 1% coming from? Because chimpanzees. Yeah, okay. All right, good. At least that ballpark is there. 0.001%. Why 0.001%? Where's that? Huh? Just speak up a bit, I can't hear.
Like it's roughly 0.00.
No, but why 0.001? Where does this number come from? So 1% is divergence between humans and chimpanzees, like an average human and average chimpanzee, right? Where does 0.001 come from? Okay, maybe we'll talk about it a bit later again. Sorry? Something like SNPs, okay. Okay, we'll come back to SNPs, yeah. Okay, so what are we talking about when we talk about alleles? You've already heard this term previously. What are alleles? Different forms of a gene. And if I have 20 different mutations that separate my form of gene X and your form of gene X, is that sufficient to describe two different alleles? No? Why not?
They should produce different phenotypes.
Is that necessary for defining alleles? No, right? So actually, even a single mutation can define two alleles, right? They're basically two forms, correct, of the same gene or the same locus, right? So alleles are how we talk about specific types of genetic variation, which is segregating in the population. Polymorphism is when there are different sets of alleles in the same population, right? At the same locus. Locus is just a position in your genome. A locus may be a gene. A locus may be a number of genes. A locus may be an operon, right? So the way the word locus is used is very flexible. It's used in very different ways, right? Without really being very specific about exactly how much of the DNA are we talking about when we say locus. What is ploidy? Sets of genes, sorry, what sets of chromosomes, right? How many copies of the chromosome you have. So how many copies of chromosomes do we have? 2, right? 1 from each parent. A typical bacterium will have a single copy of entire chromosome. There are bacteria that do have several copies. What's a genotype? Oh God, I can't hear anybody clearly. Okay, why don't you say? Oh, you. Sorry. Yes. Genetic traits. What does that mean?
Which is different from the phenotypic traits. How in the gene is the sequence that is constituting the phenotype for a certain trait.
So you want to try and be a little bit more precise, yes? Yeah, allelic combinations that you define for a set of genes, correct? So again, genotype is one of these terms which is used very, very different ways by depending on the context. So I may define a genotype based on 2 genes, right? Are you big A or little a? Or are you big B or little b? And some combination of these can determine genotypes in my study. In your study, you may take 100 genes and you say together those all encompass genotypes, right? So genotype is again to be defined every time you're discussing it quite clearly. Any confusion about any of these? Yes.
Polymorphism is an individual level concept. What's the difference? Genetic variation.
Polymorphism is saying in a given population, there are different alleles that are present at the same locus, right? So that would be polymorphism, right? Genetic variation is a catch-all term where you can, polymorphism is one way you would measure genetic variation, right? But you could also measure genetic variation in other ways. Yeah. You had a question?
That was me who
Yeah.
was seeking that clarification between alleles and polymorphism. What you explained.
Okay. All right. Anybody else? All right, good. So when we're talking about evolution, we're typically talking about changing allele frequencies over time, right? And so now that you know what's an allele, it's one variant at a particular defined locus. And we're asking over time, how does the proportion of this variant in the population change, right? And that's how we typically define evolutionary change. And a lot of what's happening in this workshop is going to be obsessed with us understanding this, predicting it, measuring it, and so on and so forth, right? Okay, so you've already heard these terms, fixation versus loss. What is fixation? What would be the allele frequency? Right. So it's fixed, which means that all copies of that particular gene are of the same form. Loss would be the opposite, where your allele frequencies are reaching 0 or close to 0. We can define mutations in different ways. I'm going to again go through some terms because you'll hear these again and again. One way to define them is what's the size and what type of mutation is occurring. Point mutations are where a single base is changing, right? If a G changes to an A, it's a point mutation. If a G changes to a G, is it a point mutation? Do you even know that a G changed to G? You don't, right? So if there is in fact an event where some base has changed, let's say G went to A, went back to G, you would not know, right? What's an insertion? Where there's a small piece of DNA that's introduced when copying DNA, for example. So if you have a string of A's and now suddenly there's a G in between that's inserted by mistake, that's an insertion. Insertions can be long, can be short. Similarly, deletions where a piece of DNA is deleted. This is frequently found to happen during replication of DNA, where there is a strand sort of loops out, and then a bunch of stuff is no longer copied correctly, right? You can have inversions where typically big, but not always, pieces of DNA can get flipped. That's when we are most interested in them because a lot of things can change when these big inversions happen. By flipping, what is changing in the DNA? Why do we care about it if there's an inversion? Sorry? It may not recombine. That's, I think, very high level stuff even, right? But very simple, basic stuff. The order, right? So the order of your bases is now changing. So where you had, and so on. This is what you get, right? And your gene is being read in this direction for making proteins. Clearly, you have a problem because the sequence no longer matches. The amino acid sequences might be different, right? So inversions have this impact as well as several other impacts, such as change in the orientation also changes expression levels sometimes, regulatory regions change, many different things. So inversions are interesting, and you'll hear, I'm sure, about inversions later on in the school. Translocations are when pieces of DNA move from one location to the other, and they will have, in some sense, similar effects to what inversions can have, right? Your local context is now different. A piece has gone missing from a place where it existed, so part of a gene is gone, maybe, part of a regulatory region is gone, and it's creating a new regulatory or coding region in some other place somewhere, right? So translocations can have potentially large impacts. Duplication, as the name suggests, is when a particular piece of DNA is copied somewhere else, right? All right, yeah. You could call them a bunch of point mutations as well, fair enough. Many times what we have is inversions and translocations, duplications are caused by specific enzymes or specific mechanisms that lead to this sort of big change en masse. But you're right that if I only had a 3-base inversion, right, it could be 3-point mutations. Yeah. Any confusion anywhere in this? All right, yes.
Doesn't all genes downstream get affected?
Yes, can get affected, right? So for example, so question is what happens to downstream genes? Will they not get affected? But that depends also on what is the structure of your set of genes in a particular region, right? So let's say this is one gene, this is another gene. Now, if a bunch of things get shifted here, it may not have an impact on this gene downstream, right? Because you're missing a piece of this, it's fine. If, however, it's clearly going to affect what's going to happen for this gene, right, for the rest of this gene, and where it's going to stop, perhaps. If it's part of an operon, where there's a single mRNA being made from this whole set of DNA, then a change here will have influence on what happens downstream as well, because it's one big mRNA that's being generated that is then translated separately. Yes?
The number of locus will always remain same, or
Number of locus and what will remain same?
like when the duplication happens, so is there, it will change the current this sequence, or it will just add to that?
It will add. Typically, we're talking of duplications. We're saying if this is my previous genome, I basically have, let's say, this chunk that gets duplicated. So my genome is now going to be bigger, right? So duplications will typically add more sequence. Sometimes duplication can happen with a bit of deletion where the insertion site is, but that's usually minor. Okay, good. We can also define different types of point mutations. You can have transitions or transversions. Transitions are between purine to purine. Or pyrimidine. Pyrimidine. And all others are transversions. You can have G to C mutations, A to T. So you can define mutations, as you can see, in many different ways, right? And which kind of mutation happens may become important for evolution. I'll talk a little bit about this later. But by and large, when you're talking about broad models in population genetics, you often may not consider the details of what kind of mutation is there, right? You're saying one mutation is there. You're broadly interested in the statistical properties, perhaps, of what are the phenotypic effects of mutations, what is the mutation rate, and so on, without bothering too much about what kind of mutation it is. Okay. But I do think it's important to understand what we're talking about when we're talking about mutations. You can also have mutations which are coding or non-coding. What are coding mutations? Which code for amino acids, right? A sequence of amino acids. So this will typically be mutations that change or could change the amino acid sequence, right? But if it occurs outside of the part of the gene which is transcribed, so if you have mutations here, for example, which does not encode a particular gene, these will be non-coding mutations, okay? Non-coding mutations are often thought to have lower impacts on phenotypes of organisms, but that's not always the case. We're discovering more and more that a lot of these non-coding regions have elements which regulate expression of the genes which are coding. And so depends on the context, what's going to happen in terms of a non-coding versus a coding mutation. Finally, within the coding region, you can have mutations which are synonymous or non-synonymous. As the name suggests, synonymous mutations are mutations that will not change the amino acid. Okay, so here is the standard genetic code. I think most of you are familiar with this, but I'll just walk you through it again. So our amino acids are encoded as triplets of bases: first base, second base, third base. This is a simple table that kind of works for most of life, which is pretty incredible, that the same code allows us to decipher what is the string of amino acids that any organism's DNA is likely to give rise to, right? That's pretty amazing. Okay, so here's how you read it. The first base is U. This is on the mRNA. Is everybody okay with what is mRNA, or should I? Yeah? Sorry? Okay. So on the mRNA, the first base can be U, the second base can be U, and the third base can be A, and that encodes for leucine. Now, if you have a A to G mutation in this position, you're still going to get leucine. But if you have a UUA to where's the one? UC mutation, you're going to have a phenylalanine, right? So that's a non-synonymous change. Why do we care about this? Because synonymous changes which don't change the amino acid should typically not have a big impact on the protein and on phenotype, whereas mutations that do change the amino acid are more likely to have an impact on how the protein is functioning, right? Or how the protein is formed. All right, this you have already seen. So all the evolutionary forces that were listed earlier, there are a bunch of them that are going to do the job primarily of introducing variation, right? So migration means that new alleles may enter a population or may leave a population. Mutation is going to introduce variation always. Recombination is also going to generate new genotypes, right? So remember that with recombination, you're not necessarily generating entirely new variation. You're reshuffling the genetic variation that existed. Perhaps in different individuals, it comes together, in different chromosomes, it comes together, and so on. So these are all situations where variation is going to increase in a population. Yeah. I guess yeah, you need selection to act with it. True. No, not by itself. I guess it can generate new alleles though, if recombination is happening. Yeah. Okay. Natural selection and drift are going to decide what's the fate of that variation, right? So if you have large populations, natural selection is more likely to determine what happens to alleles. If you have smaller populations, they're going to be more stochastic, and the fate is not going to be as easy to predict. We can define selection in many different ways. Yes. It doesn't sort variation in the sense it doesn't decide the fate of the variation. No, if you have new migrants coming into a population that carry new alleles, which don't exist in this population, right? The effect is going to be, depending on the frequency, like mutation, a new mutation came in. Yes, yes. Okay, selection can be of different types. We call positive selection in a case where an allele is beneficial, right? It will increase over time in a population. Negative selection is the opposite, where an allele is deleterious, it is undergoing, it's going to be purged out of the population. Purifying selection is defined for a particular location, where at that location in the genome, if any variant comes up, then it is deleterious. Then it would mean that that existing variant probably stays, okay? Other variants are going to all be removed. So purifying selection is defined at that site, okay? Whereas positive and negative selection general terms used to say what kind of selection is acting in a population at a particular phenotype, for example, for a particular phenotype. Okay, you can have other kinds of selection that are defined. Again, you'll hear much more about this in detail later. Balancing selection is where you have different kinds of selection pressures acting to determine the fate of an allele at the same time. You can have, for example, frequency-dependent selection, where whether an allele is beneficial or deleterious is determined by its frequency in the population. So some alleles at very high frequency may be beneficial or deleterious at high frequencies, and then their frequency can reduce over time. You can also have heterozygote advantage, where a heterozygote in a diploid population, for example, individuals with this genotype may have maximum fitness, whereas these have lower fitness. In this case, the selection acting on each of these alleles is determined by which genotype context they're in, right? Big A is beneficial when present with little a, but not necessarily by itself, right, in two copies. And finally, you can also have spatial or temporal heterogeneity in selection. So if you have a population, let's say, which is occupying some altitudinal gradient along a mountain, a particular allele may be more beneficial here, but less beneficial elsewhere, right? So so that means that when you look at a whole population level, you're not going to be able to generalize what is the kind of selection acting on a particular allele, because it really depends on where that allele is located in space or similarly in time. Okay, this I'm sure everybody is familiar with already. Stabilizing, directional, and disruptive selection. Which of these is going to reduce genetic variation in a population? Sorry? Directional selection. Why not stabilizing selection? So with directional selection, we're saying, for example, that only these individuals in the population are more likely to reproduce, right, successfully. So it means that this variation is going to reduce over time. What about stabilizing selection? Sorry? So for example, if this one has the maximum fitness in this population, then this variation should reduce over time, right? Individuals with this or this phenotype are both less fit compared to this phenotype. And finally, disruptive selection is going to have the opposite effect, where you may eventually end up with a situation where the phenotype distribution may be bimodal, right? Because intermediate phenotypes are most deleterious. Yes? What is the relationship between balancing and stabilizing? So with stabilizing selection, we're typically thinking about one form of selection acting on a particular phenotype, for example. With balancing, we're often talking about a particular allele or a locus facing different kinds of selection. And those selection pressures may come from different actual phenotypes that could be influencing what's going on. Mike, are you going to talk about balancing selection more? Okay, I will send you literature then to read. Any other issues? Did I answer your question? No, it's not most. I mean, they will both reduce variation over time. Yeah. And really the strength of selection will determine how much variation is lost, right, over time. Any other confusion? All right. Finally, these terms you'll hear again is convergent and divergent evolution. Anybody have any idea already what convergent versus divergent evolution means? Or selection? Sorry, I can't hear you very clearly.
Like they are like evolving in different environments, but they are evolved for same like phenotype which is like beneficial for.
Same phenotype. Okay, so selection is leading to phenotypes looking similar.
Yes.
Is that what you're saying? Yeah.
How that may have come about. So it may be because basically they are evolved from different ancestral groups. They look similar, they have similar phenotypes because of environment.
So convergent evolution is actually used in many different ways, slightly different ways, so that it's confusing. And yes, sometimes it's meant in the way you're talking about, where the genetic mechanisms that give rise to a particular phenotype are different, but they have converged on a similar phenotype. It can also be used to talk about different environments leading to similar phenotypes, both getting selected at, in similar ways, so that again the phenotype is looking similar. Did you have a question?
Can we say here that when two sequences start and then some selection adds something to the same would make the phenotype eventually, but not to that.
So people use that terminology as well to talk about sequence convergence, right? Which is why I was saying it's another of these terms that is used in slightly different ways in different contexts. Important to define what you mean. Divergent is a little bit easier, right? Because it's more straightforward. Things are different. So there are different selection pressures acting on, let's say, two populations. They start out perhaps identical, part of the same population that gets split. And if they're facing different selection pressures, they are likely to evolve in different phenotypic directions, right? And similarly, if you have different selection pressures acting on different populations which have different ancestry, they will, of course, also are likely to go in different directions when it comes to phenotype as well as genotype. All right, I think those are all the major terms. You have seen some discussion of fitness already, but we'll often define for diploids or haploids fitness depending on each genotype. So for diploids, you will define fitness of each genotype independently, right? We're typically interested in relative fitness when we're talking about evolution, not absolute fitness. But can somebody give me an example of how I might define absolute fitness for a plant that I'm studying? Number of seeds produced by the plant, okay? And what would be an example of relative fitness? I have a bunch of plants here. Sorry? Why can't we stick to seeds? Okay, so you can calculate relative fitness with seeds as well, correct? But why do we say relative? We're comparing different genotypes to each other, right? And so we'll set one genotype as our reference point and then calculate everybody else's fitness according to it. So reference point will have a fitness of 1, and the other genotypes can have different fitness defined, right? And the reason we're interested typically in relative fitness, why not absolute fitness? It seems like a great idea if I produce 100 seeds versus 10 seeds. We get a? No, but the genotype is of the individual, right? So we do care what an individual does ultimately. That's what's going to lead to population level dynamics. Why are we not as interested in absolute fitness?
Because there is no meaning of considering.
Okay, that's a good way, yeah. We don't know the threshold fitness. Huh. No, that's what you're saying, but you do need to look at each individual, right? To understand what's the average genotype fitness like. Yeah.
When you are looking at relative fitness, not just the number of seeds, but also need to see how many of those seeds succeed to produce new. So if you have a lot of seeds, but only some of them are becoming contributing to the next generation, then it's not really that helpful. So if you can include that factor as well by looking at two plants, different number of seeds, but also their offspring.
So if you go back to when people were first starting to think about, I think all of you have elements that are right and some confusion. So you've heard of Malthusian fitness, right? What is Malthusian fitness? What is Malthusian growth, population growth? Right. So the core idea of why, how natural selection might act, the most intuitive, simple idea is that there's competition for resources, right? That everybody cannot survive. Not all offspring that anybody may produce are going to be able to survive. So given that there is some competition, given that not everybody is going to have a chance to make it, relative fitness matters a lot more than absolute fitness, right? Of course, if your absolute fitness is 0, you're not producing any offspring, then you're toast, right? Then your absolute fitness is 0, and so is your relative fitness. But even if you're producing 2 offspring, if you are the only one that produces 2 offspring and everybody else typically produces 1 offspring, you are going to have an advantage, right? And so it may not be great, right? Your population is not going to go booming around perhaps with producing 2 offspring, because let's say one of them is doomed to die. But everybody who produces 1 offspring is in even more difficult situations, right? Compared to, does that make sense? So intuitively, that's why relative fitness is much more important often to think about, even though absolute fitness may be important in certain conditions, okay? All right, so when we are defining relative fitness, we will talk about selection coefficients a lot. This is typically denoted S, and what we're using S is to effectively compare our different genotypes. So for a haploid genome, if I make it simpler, I have big A and little a as my 2 genotypes, right? Imagine in a bacterium with a single copy of a gene. If WA is big A is my reference, which is 1, and W little a is 0.9, then the selection coefficient against little a is 0.1, right? So this would mean that little a allele has lower fitness compared to big A allele, and the selection coefficient is a measure of the magnitude of that difference in fitness. Okay, so I would like you to think a little bit about how much variation do populations harbor and how can we measure this? So already we said that the amount of polymorphism in a population may be a good measure of, how much genetic variation there is. How else could we estimate genetic variation in a population? And how much variation actually exists in populations? So probably many of you have heard vaguely, randomly that, okay, humans in India have more diversity, right? What is that based on? How do we know this? How do we guess this? In terms of phenotypic heterogeneity, is that what you're saying? Yeah, that could be one measure, but of course there are problems with that, and we'll come to that a little bit later. But so you might be able to measure phenotype, thinking there's some underlying genetic variation that may give rise to that phenotypic variation, right? What other things could you do to measure? Sorry? Change in allele frequencies. How will that help estimate genetic variation? More allele frequency change, more could be variation. Not necessarily, no? Why?
That's the base of variation. The basis of having more variation.
You're saying if allele frequencies change, that means there was more variation to begin with. But that's going to be useful in comparing, let's say, within a population across 2 different loci, correct? But across populations, you can't compare allele frequency change as an indicator of genetic variation. Is that making sense? Okay. Yeah, so you could sample individuals from your population And what are you going to measure? So polymorphism is one thing, right? You can say that these are sites or loci or genes that I care about. And I will measure polymorphism at these sites across the sample that I've collected. Is there something else you could do? There we're defining some sites that we're going to look at, right, for polymorphism measurement. Mm-hmm. Yeah, good. So this is similar to the diversity indices that ecologists will use, right? Saying each allele that I'm finding in my polymorphism data set, what frequency is it at, right? Are these very, very, very rare alleles, lots of them? Or are there fewer, bigger alleles? And maybe that means something different for the dynamics of the population or the genotypes and so on. So the reason I put up this question is this is how a lot of people in evolutionary biology many decades ago, when we first started understanding the nature of the genetic code, but trying to understand how much variation exists in populations, right? Because if selection is very strong and it keeps eroding variation many times, right, in populations, you would expect to see very little genetic variation. And yet, when people started measuring different kinds of genetic variation using allozyme markers, using sequence polymorphism later on, they kept finding a lot of variation. And so this became a question that was a big question to answer. How is this variation existing? Why is it still around? Right? So this is what thinking about. The other question is how much of this variation is relevant for adaptation, right? So a lot of us will have lots of variability, right? Comparing our pairwise genomes, let's say. But much of that variation may or may not be relevant to determine our fitness. This is true for any population. Why is that the case? So synonymous mutations, right, may not be, its variation counts as variation, but may not have a big impact on phenotype. All right, sorry, you were saying? Correct, right, so the same thing. What else? If it's non-coding, like it may not confer big phenotypic differences, maybe, maybe not. We come to that, yeah. Yes, right. So there is another layer which is important to think about, that just because the genotype gives rise to a phenotypic difference doesn't mean it's relevant for selection or adaptation at all, right? If there is no selection for height in humans, it doesn't matter how much variation there is in height, which is genetically encoded, it's not going to be under selection, right? It's not going to lead to any adaptation. So there are two layers then to think about. One is whether your variant leads to phenotypic change, and second is whether your phenotypic change leads to any fitness consequences or not. Okay, so does selection see the phenotype or the genotype? This is easy. Sorry? Phenotype, yes. Anybody who wants to bat for genotype? You want to bat for genotype? Okay, why? How? Hmm. 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 I see your logic, but the problem is that how will natural selection sort of know, right, what is the genotype of an organism? The only way selection can act on it is through phenotype, right? Does that make sense to everybody, right? Yes.
For example, if there are injected versions which might not make the organism different, but still a selection would work.
So she's suggesting that maybe inversions may still work that way. But remember, what do we mean when we say somebody has higher fitness or lower fitness? Ultimately, reproductive fitness, right? You've got to survive, you've got to reproduce. So if your inversion has no impact on either of those two, then selection cannot act on that inversion directly, right? You had your hand up? Yeah.
There are dominant mutations.
Mm-hmm.
Selection pressure.
Mm-hmm.
Same on cognitive
Mm-hmm.
Is that why like the phenotype becomes more?
Not why. there's no way for the genotype to be more critical than the phenotype in these situations, right? Because unless having an A there somehow makes it more likely to propagate without having any impact on your reproduction or survival. Yeah. So you're right that because heterozygotes, if they have same phenotype as homozygous dominant, then the recessive allele gets a free pass along with the heterozygote, right? And so there are cases, of course, we know of where heterozygotes persist in the population, or the recessive allele persists in the population because of this association whenever it's in a heterozygote form. So that shields it from selection itself. But that's a different, right? That's still through the phenotype. Because it's the phenotype of the heterozygote which is similar to the phenotype of the homozygote dominant. Yes? Mm-hmm. It's going to be different. Yes, it will be different, but selection will still have a harder time getting rid of the recessive deleterious allele, simply because it keeps getting generated through assortative mating. Make sense? Oh, sorry, not assortative mating, through mating, random mating. Okay? All clear? All right, good. Yeah.
Last point.
Ah.
As you said, selection only sees fitness, the organism, the reproductive success. And I believe that's different from phenotype.
Yeah, the additional layer, yes, you're right. I'm equating phenotype with some measure of fitness here, yes. But it has to be some phenotype. The point is that your reproductive success,
okay, it's a phenotype. No.
It is a phenotype, right? Effectively.
Okay, then I.
Okay?
Okay, so I always think phenotype.
And then fitness, correct. But my point is that you can't go from genotype to fitness directly.
Yes.
You have to go through phenotype.
Yes, but neither genotype and phenotype as well. I mean, in the end, it matters fitness, right? I mean, for competition.
Yes. But it is phenotype that leads to fitness, that decides fitness, right? Okay. Yes.
There is some concept of gene selection.
When what? Non-random mating may happen in several contexts.
Meetings are now dependent on the phenotype is actually causing that phenotype to be different from the other thing. So is it selection seeing the phenotype directly?
No, sorry. Think about it a bit more, right? So you have genotype A, genotype B, okay? And let's say they have different colors. What determines mating? Colors, right? So you have to have phenotype come in between. Yeah.
There is sexual dimorphism, so it completely depends on the phenotype whether the mating will be successful or not. So it will determine the reproductive fitness of the animal.
Yep.
So there are also phenotype functions.
Always. You need, right? Because there's no magic microscope that can be used to look at the genotype directly. I don't know how else to.
But at the same time, I mean, phenotype is a well-defined object, right? I mean, phenotype in principle can be infinite phenotype. So, I mean, we usually try to make sense of what makes sense fast. Like, however, genotype is the one which is well-defined. It's another thing that cannot be accessed in the current state of the
It's a problem, I agree. So it's much easier to define genotypes than phenotypes, right? Yes. But the fact remains that reproduction is a very phenotypic thing. Yes.
But the thing is, the phenotype should become a phenotype. One time there must be some mutation.
Yeah.
So first time
Yeah, may or may not, right? So in a specific case, if you want to consider that there is complete nice association between genotype and phenotype, where there's one mutation that gives you phenotype A, right? And a different allele that gives you phenotype B, then every time selection is acting on the phenotype, effectively the genotype is what is changing, correct? But the point is that it's not directly the genotype. And this is important to get sort of comfortable with, because otherwise it leads to problems later on. Yes. Yes, what if the phenotype is plastic? And which is precisely where the issues come in, right? The connection between genotype and phenotype are not one-to-one connections, right? Which is why every time selection is acting through some phenotype, it doesn't mean that genotypic change is necessarily going to be commensurate with that, because it really depends on what is that connection. Which is why like a substantial proportion of people in evolutionary biology are trying to understand this genotype and phenotype connection, understand the phenotype and fitness connection, right? Because without it, it's very hard to predict what's going to happen. No, fitness is its own thing, right? Fitness is defined by reproduction, for example, survival, but it is phenotype which ultimately is the traits that lead to survival higher or lower, right? Or reproduction higher or lower. Because of all the various problems with the connection between genotype and phenotype, it's not genotype directly, which is being seen. All right? Okay. Okay, good. All right, so I'd like you to go to this population genetics simulator on your laptops. The link is in Slack as well in my channel. And what this simulator is a simple way to start gaining some intuition about what happens in populations. Okay, so you have red and blue genotypes and phenotypes in this, if I remember correctly. And you have alleles. You have the ability to dial up and down the selection pressure, the selection against or favoring each allele. You can have heterozygote advantage or not. You can change your population size. You can simulate for as many times. I think there's some limit because it's a web server. Those of you who don't have laptops, just share with your neighbor, right? Everybody found it? Yeah? Got the basics? Okay, so then I'd like you to think about what are 3 ways you can make the red allele fix. Okay, try. You can discuss with your neighbor if you like. Given that you've already gone through Kavita's lecture, this should be fairly easy. Anybody succeed? Got it? Oh, your red allele is dying. Yeah. Yes. Got it? Okay. Tell me what's one way?
Survival if we modify for the other.
Uh-huh.
So what I did was partial by 9 by 9.
Sorry?
For the other two, the blue and the purple, when it
Huh.
reduces the survival. But it still has marginally more than that over time. Can happen.
Good. So what he's done so far is increase the survival advantage for the red allele compared to the other two genotypes, purple and blue. And that allows the red allele to be fixed. Yes. Okay. Are you cheating? You're doing two different things. You've got a survival advantage and a mutation rate advantage, but good enough. Yeah. So that's a second way that you can make the red allele fix, right? That you basically make other alleles mutate to the red allele. What's the third way? Anybody? Sorry? Yeah.
Third way is start with a very small population and hope that stochastic.
Hope? No. You got to make it fix. There's no hoping. If the red allele is not fixed, we're all going to die. Some such thing. Okay, anybody else? Somebody who's been very quiet so far? You know, I'm looking at you, yes. Increase the starting frequency. Yeah, good, right? So if you have a higher frequency to begin with, you're more likely to go to fixation, even in a relatively smaller population size. Sorry? Yeah. Yeah, that she did already. Okay, good. Is everybody okay with this? Yeah, getting this to work? All right, another question. Set the survival rate so that there's a heterozygote advantage, which means the purple is going to have a higher survival. What happens to the red and the blue alleles? What do you expect will happen to the red and the blue alleles? Huh? 0.5, 0.5. They will have similar frequencies. Is that what everybody else thinks? Will they necessarily have similar frequencies? Yes? Always? Okay. So he's saying that if your heterozygote advantage is high, the red and the blue alleles will always have the same frequency. Is that what everybody's finding and expecting? Yeah? Okay. Can you change that? Is there a way to make it different? No way to make it different? Yeah. So one way is to introduce more drift, right? Reduce your population size so that despite a heterozygote advantage, both alleles are not necessarily always going to be at the same frequency. Anything else? Yes. Right? So you can still have heterozygote advantage, but not see both alleles at the same frequencies. If one of them is slightly better than the other. Are you seeing that? Try it out. Okay. Then the last question. Set the blue allele to have a selective disadvantage. What would you change to prevent its loss? You got it to not be gone? Mutation. Okay. Mutation what? Mutation rate higher, yeah. Increase population size. Decrease generation is a little bit cheating, but anyway, yeah, sure. I mean, if you just do it for 2 generations, it may not be lost yet, but yeah. Okay. But if you increase population size and blue has a disadvantage, it will be lost, right? That's not going to help. You cheating? No. Cheating is not allowed. What else? Red to blue mutation rate can increase. Anything else? Decrease? Technically not the same, but yes, fair enough, similar. Okay, so play with it a bit more afterwards in the evening and see what you find. This is a very nice tool to get some intuition about what you expect to see. Does it actually match up? Sometimes our intuition is wrong. We think that certain things should happen. You run the simulation and it doesn't work. So it's very useful to think about stuff. So do use it more. Okay, so I think what you effectively seen in some of these exercises that you just did is that the population size and the selection coefficient together determine fixation probability and dynamics, right? So only population size being larger or only selection coefficient being very high or fitness being very high is not sufficient to determine, to be able to say what is the probability of fixation of the allele. So in general, at small population sizes, drift dominates. You've seen this. And selection coefficient has to be large enough for a beneficial allele to be able to fix rapidly, right? If you give a slight benefit to one of the alleles, even with a large population size, it's going to be harder. But with small population size, it's very hard to fix beneficial alleles, despite a substantial advantage. At large n, selection will dominate, and even a weakly beneficial allele can go to fixation much more easily and much faster. Finally, at large n, not all alleles behave as if they're evolving under selection, right? So neutral alleles will behave still as if they're evolving under drift, even with a large population size. Does that make sense to people? So you can try in your simulations, set both alleles to have same fitness, right? Effectively, these are neutral alleles then, there's no difference in fitness, and see what happens even in relatively large populations. They will behave as if they're evolving under drift. So this idea that population size and selection coefficient together determine a lot of what's going on is important, and you'll see the maths for this in the subsequent lectures much more. But I think it's useful to have some of these general broad conclusions in your head so you can think about data sets and your own results much more easily. All right, I want to switch gears now to talk about what's the phenotypic effect of mutations and how do we get to this kind of answer, because this is something clearly that's important for us to understand, right? What is the genotype-phenotype connection? This is what we talked about. One way to get at this is to take individual mutations and measure what impact they have on fitness, right? So this is what's called the distribution of fitness effects, the DFE. And this is basically a histogram depicting what is the statistical distribution of consequences of single mutations on fitness. So the first set of nice studies showing this was with viruses. These are data from vesicular stomatitis virus, infects plants. And what they've done is they've generated all these mutant viruses using mutagenesis and then passaged them in the plant and asked, how good is this virus at propagating and infecting the plant? Because they can measure how much virus there is without too much difficulty. So what you see here is on the x-axis, a fitness of 1 would indicate neutral mutations, right? These are all mutations in the virus that did not impact its ability to infect the host compared to the wild type earlier. Remember, this is all relative fitness that we're talking about, right? So we're taking our reference original virus, our wild type virus, making mutations and asking what effect does those mutations have. And to the right here are beneficial mutations. What is the selection coefficient for this one? 1.1. Anybody? 0.1. So it's basically improving fitness by about 10%. What about these guys? What are these? No effect. No effect is here, 1. Okay, so what are these? Dead, right? These are dead. So a lot of lethal mutations in this particular virus, right? Is that surprising? Is anybody surprised that there are so many lethal mutations? Or does it seem reasonable? Nah. Okay, all right. I thought it was a bit surprising when I first saw this. How come there are so many lethal mutations? Because it suggests that this virus is sort of teetering on the edge of, right? Very dangerous life it's living. A lot of single mutations can drive it extinct, right? Yeah. Yeah, so in a real population, they will be selected out, right? I mean, as in they're dead. But in this experiment, because they allowed the virus to have the chance to infect the host, and basically what the reason they're saying it's 0 is because it didn't manage to infect, right? But they knew that the infection was successful, like they did everything right, so that if the virus was able to infect, they should have seen it, right? So the opportunity was given to the virus, but then it was dead. Okay, so so as I was saying that I was surprised that there were so many lethal mutations in this virus. Do you expect to see similar distributions for other organisms? Any ideas? Humans, bacteria, plants? Stunned silence. Okay. Come on, any ideas? Can you make no guesses at all? You guys, half of you at least are biologists. I know this. You know some biology. Less the lethal mutation, more the complex. Okay. What are complex organisms? Okay. And why do you say complexity is important?
Because there are like certain mechanisms in more complex or easier to balance out.
So you're saying there's buffering, there's some redundancy perhaps, and so on. That's a good argument. Any other arguments?
Organisms with higher quality will have lesser.
Yeah, that's a good answer, right? Because there are, again, buffers and redundancies built in because of ploidy, perhaps. Any other? See, you should have ideas, right? There's nothing wrong with having ideas based on whatever you do know. And you already know a lot to be able to make guesses. Yes?
It also depends, right, how evolved the strain. I mean, if it's close to the peak, then most of the mutations are going to be eliminated. But if it's somebody which is kind of just crossed like at the valley, then it could be that new mutations are mostly beneficial.
Right. So, but your answer is not really about organisms, right? Comparing across organisms.
So
but that's a good answer.
Going down, so probably that. But I think it's the landscape.
You know what you're talking about in general, so that's fine. But yeah, anybody else? Yeah.
Do viruses also follow the same codon chart?
Yeah, some of them have slight differences, but by and large, yes.
And shouldn't the frequency for 1 be 0.3?
Frequency for 1 be 0.33, why?
Because of synonymous mutations.
That's a great hypothesis, yes. I think I'll touch upon this a bit next time when I speak. But yes, your intuition is valid. Anybody else want to take a guess? Would you see similar DFEs for other organisms? So there's one answer about complexity, one answer about ploidy. Anything else that might be relevant? Proportion of coding versus non-coding. So with viruses, I think, and others also think, I think some of them others, that because the genome is so streamlined, right? The genome is so condensed, maybe that leads to a higher proportion of lethal mutations, right? And that's similar to what Shazi was saying, that the proportion of coding versus non-coding or something like that is an indicator of what is packed, how much information is packed into the genome. So, okay, so how do you model the DFE? This is again of interest to a lot of models in population genetics. So this paper I've put up in Slack, you should read it if you're interested in this more. And the general idea is you want to get some idea of what the shape of this DFE looks like, because then you can make the maths simpler, easier, more workable, etc., right? And so people have, for example, used a gamma distribution whose shape you can change using these parameters to have these different values. And according to, you can see it looks very different, right? So the distribution is the same class of distribution, which means you can work with it in a similar way, but the outcomes are quite different in terms of what the DFE is going to look like. How fat is this tail of beneficial mutations? How many mutations are deleterious, and so on and so forth, right? So this is more by way of wanting to introduce some of this terminology to you, so you have some understanding of what one does with this. In adaptive evolution, we typically think that this tail end, which is the beneficial mutations of the DFE, are involved, and that's going to drive adaptation over time, right? This makes sense. So here is what's called extreme value theory. I think you'll hear more about this later on, which is what is used to make predictions or ask how will populations evolve under selection, where you make assumptions about the form of this tail end, which is the beneficial mutations, and use it to say, okay, fine, for example, if it's exponential, there should be a certain way that these beneficial mutations will drive adaptations. What is the time that you have to wait for how long you have to wait until a beneficial mutation will come, how long will until it fixes, how long until fitness increases, and so on and so forth. So this is what these DFEs can be used for. And empirically measuring the DFE is important. So here's an example in contrast to what you saw with the virus data set from one protein, ribosomal protein subunit in Salmonella, a bacterium. And here you find that there are almost no beneficial mutations at all, right? So the DFEs are shaped quite differently from what you saw in the virus. There are also no lethal mutations to be found here. There are some mutations which are very deleterious, right? So fitness is 0.7, relative fitness is 0.7. But there are no lethal mutations to be found here. Any guesses why? There are no lethal mutations that they could observe in these experiments. They don't survive. So in this experiment, unlike viruses, where you know that you put in the viral particle, right? In these kinds of experiments, you don't know whether the mutant was successfully made, which was lethal, or it was, because you don't see it, right? So if there are lethal mutations, the only way you can detect them is if you test the DFE, test the fitness in a different environment, right? So you have a colony growing on some medium X in your agar plate, and there, of course, it's not lethal because it's growing. And then you put it in some other medium, have it grow, and then if it doesn't grow, then you know that the mutation is lethal in that environment. Make sense? So that was not done here, so we don't have lethal mutations. But you can see there are a lot of deleterious mutations, and most mutations here are deleterious. And that matches with the idea that people have had that many, many mutations, by and large, are going to be deleterious. Yes? These we know because they grew, right? Yeah, they grew, but just not as well as the ancestral wild type. Okay. Any guesses what this DFE would look like? So this is a ribosomal protein. Ribosomal proteins are rather important for cells to make proteins. Any guesses what we might find a similar DFE looking like if we took another protein in the cell? A different type of protein. Very similar or very different? Similar? So you'd expect that the DFE is going to be, you expect that proteins have evolved over time, right? For such an essential and important protein, that the nice beneficial mutations have already been fixed in this population, probably, likely, right? So many more mutations are going to be deleterious. But for another protein, let's say, which has acquired a new function, okay, in evolution, you might be able to find many more beneficial mutations depending on that protein's function. Okay, ploidy changes are really dramatic or can be really dramatic in terms of phenotypic effects. So technically, it's a single mutation if you have a ploidy change, right? There's a duplication of the whole genome, which has happened a long time ago in yeast, for example. That's one mutation, but the whole genome has now 2 copies, right? Yes?
You are saying that it is not a what type of protein
I'm not understanding your question.
I'm just like, did you say like an protein, what is
I'm saying that you may be able to find more beneficial mutations if this is a protein that has recently evolved a new function. That would be my guess, right? But I'm saying that it's worth trying to think about this and make some guesses as you go along, because you will not always find data for everything, but it helps to hone your intuition about what things should look like. Okay, so here are examples of where a ploidy change from a diploid to a tetraploid has really large effects in these plants, right? The flower size, the leaf rosette shape, size, everything is increasing, changing. There's another example that I'm sure all of you are aware of with Down syndrome in humans. If there's trisomy for one particular chromosome, there's a really large impact on phenotype, also on fitness. So ploidy is one way where a single mutation can have a really big impact on fitness and phenotype. And so ploidy is something that people are very interested in, how it evolves, what are its impacts, and so on. Okay, I want to go through a quick primer for what kinds of evidence do we use to understand whether selection is going on or not. This is from something where I was wanting to talk about synonymous mutations. So ignore the pink stuff in here, which is about synonymous mutations. I'll come to it later. But here's just some ideas of what kind of evidence people have used in general to say, detect cases where things are under selection. So here's a situation where people have been asking, for example, whether tRNA abundances and relative codon usage, which codons are used to encode a particular amino acid, could they be evolving under selection? And the idea was that tRNA genes, which are genes that bring the amino acid, recognize the mRNA, which recognize the codon on the mRNA. There should be a match between them. So in general, you expect that tRNAs, which are more abundant in the cell, should have codons, which are also more abundantly used in the cell. So positive correlation here, where each dot can represent a species or a strain. This is the kind of comparative evidence that can suggest that these are traits which are evolving under selection. Does that make sense? That if there are two things that have to work together to produce a phenotypic trait which is under selection, you expect that those two things should show correlations in gene copy number, in expression levels, in phenotypic effects across species. You can also look at the rate of substitutions, so the rate at which fixed mutations are observed in different sites. So for example, here's a case where typically synonymous substitutions or non-synonymous substitutions will have there'll be fewer non-synonymous substitutions in highly expressed genes, okay? With the idea that we know that highly expressed genes can be really under strong selection, right? We expect they should be under strong selection. So if substitutions are deleterious by and large, then you should see fewer substitutions in highly expressed genes. This can be one way to think about a signature of selection. You can think of site frequency spectra. You can look at a population, right? Like we said earlier, how are we going to measure genetic variation in a population? You can measure different alleles and their frequencies in the population. And you can ask, here's my neutral expectation. If nothing else is going on, given the population size, given that all alleles are neutral, I might expect that there'll be some alleles which are at high frequency, but most alleles are at low frequency, right? Because they just arose, they're kind of bouncing around by drift, they're neutral alleles. Now, relative to that, if I find that some alleles are at higher frequencies, that might suggest that maybe these are alleles which are under positive selection. Does that make sense to people? Yeah? Okay. You can do genome-wide association studies. So again, you're looking at the same piece of DNA across different individuals in a population. And you're mapping out all the variants that you see here, focus on the gray. And you can ask then, associated with the presence of a gray allele or not, do I see a difference in phenotype? This is what is called GWAS, or genome-wide association. You may be interested in a particular disease in a human population, for example, and that's what GWAS studies do. How do people discover what alleles may underlie a particular disease in a human population, for example? You can also have genotypic evolution in the laboratory or outside where you do experiments. You can start with ancestral populations, evolve them under some selection pressure. And if you find that some allele frequencies are increasing in these evolving populations in your experiment, then there's a suggestion that these might be under positive selection. Make sense? Okay. Finally, there can be much more direct evidence where you can generate specific mutations, right? Make specific alleles that distinguish different individuals that carry those alleles, and then ask what are the fitness values looking like, right? This is similar to the DFE experiment that we just saw, where there were single mutations in the virus, they knew what the mutations were, and they asked what is the impact on fitness. You can then generate DFEs, and at the DFE level, you can similarly ask what are the selection coefficients like, what are these alleles which should be under positive selection because they're beneficial. Here are the alleles that should be in a negative selection because they're deleterious in a population. We can identify them, think about them. And finally, you can also observe during evolution experiments in the lab or outside that specific alleles increase in frequency. You can then genetically make those alleles, right, in an ancestral background, what we've done here, and then ask now, as a result of changing that one mutation or allele, what happens to fitness, right? And if your fitness of the evolved population matches that of the genetically manipulated one, then that suggests that that is the beneficial allele that drove adaptation, and that was under selection. So this is just as a primer to say, this is how we different ways in which we can think about asking how much and whether selection is acting on variants of interest. Yeah. The second one. Yeah, so the general expectation is that genes that are highly expressed should be under stronger selection because they produce more product in the cell, and it has the opportunity to have a bigger impact on phenotype and fitness. So if we find, so the expectation is that highly expressed genes, for example, should have fewer substitutions, right? If the DFE is skewed towards more deleterious mutations, then many more substitutions are going to be deleterious, right? Which means that in general in the population, you should not find a lot of substitutions, less polymorphism. And if you see that, then that suggests that, okay, this is indeed something under selection. Okay. Yeah, so this, like I said, this slide was made for talking about selection on synonymous substitutions, but I want to just use the pictures, so ignore the synonymous part of everything here. Sorry. I was lazy and I didn't make the non-synonymous version of this. Okay, any other? So these are again things that you'll come across in research seminars in particular in this school, because you will listen to people actually using these concepts to do their work and talk about what is under selection. Okay, here are some general patterns, broad patterns of what is known of mutational effects that can drive differences in species. So the patterns of differentiation across species, right, closely related species, where we are interested in asking how speciation has occurred, what types of alleles have perhaps led to these different species diverging from each other. The patterns are all over the place. Yeah. That's a problem in GWAS in particular, right? So the question is, I don't know if you can hear the question, but the question is, many alleles may influence a particular trait. So how do we know that, how do we assign a relationship between that allele and that trait? It's hard, depending on what kind of data you have access to, right? So if you're working with experimental populations, you may be able to sequence and figure out how many differences there are between individuals. But in a GWAS type of study where you're going out and sampling individuals and asking what is the genotype and asking what's the phenotype and looking for a correlation between those two, this is a big problem. There are some statistical tools that people have developed to try and, narrow down the set of alleles which might be important for driving the phenotype, but it is not easy. Yeah. And we often cannot. So most of the time, if you look at studies that try to find alleles that are responsible for a particular genetic disease, they will have confidence intervals. They will say, okay, these may be likely, these explain that much of the variation in the phenotype, and very often it's not a very large proportion of the variation, right? So most of the time we're finding alleles that make some contribution to that phenotype, but don't explain it completely. All right. Okay, so here are examples of what people have seen. In aphids, as if you look at different species of aphids, there's a lot of small differences across the genome, right? You can, in contrast, in lizards, you have big bursts of differentiation, whereas a lizard species has diverged and split into several species, daughter species, there is a lot of difference that comes up. Then again, there may be a long period where there isn't too much change that is accumulated. The genetic architecture is quite different depending on the species. Color pattern in butterflies is driven by typically a few genes of very large effect, right? So where would these genes lie in the DFE? Towards the right or the left? The right, right? That tail end, remember, that can drive adaptation. Courtship song in cricket is something else. I forgot actually what the genetic architecture of this is. Somebody, do you remember? Okay. Stickleback have also been studied quite a while, where there are a few different genes that are known to be important for changing the armoring, as these stickleback came from marine environments to freshwater environments. Flowering time in maize is another case where multiple loci of tiny effects were involved in the domestication event. And then, yeah, similarly, few genomic peaks in crows and lots of differences in these headings, right? So basically, it isn't that differences across species are driven by one kind of few alleles of big effect or lots of alleles of small effect. It really, we see all types of examples. I don't know if we have time to go through this, but we'll go through this. Okay. Recombination. This is kind of textbook material, so I will post this on Slack again, and you can read more about it. But I basically want to talk a little bit about recombination because you need to have some visual of what recombination looks like. This is exactly how our chromosomes look like. They're red or they're blue. Just kidding. And in recombination, you're basically going to end up exchanging some piece of the chromosome. It may not be at the end necessarily. It may be in the middle, right? And these are called crossover events, where pieces of homologous chromosomes, so one that came from the mother's copy and the father's copy, they may be exchanged. And what that leads to is different ordering of genes or alleles on this chromosome with downstream consequences for fitness or phenotype potentially. So this is where recombination, this is why recombination is important, because you end up getting different combinations of your genotypes. Okay, and then the rate of this recombination varies widely across taxa. This therefore also becomes something important to consider in population genetics. As we said, recombination can generate new variation or new shuffle new variation. So if you have a very high rate of recombination, then you are going to have a lot of variability generated in the population, which should have consequences for evolution, different from situations like conifers, where recombination is measured as being very, very less, right? So how variability is generated can also differ in different organisms quite a lot, whether it is primarily occurring through mutations or recombination or some mix of these two things. Here's a nice example of experiments where people asked with yeast that were allowed to have sex versus not. Here the idea is that if you have sexually reproducing species, there's a chance for recombination to occur. And they find that total fitness increase is higher in the populations that were allowed to have sex and recombination compared to those that were not allowed to do this, right? All right. And then finally, what else generates phenotypic variation? So this is coming back to somebody else asking about this earlier. There's this concept of plasticity that I'm sure many of you have heard about. What this is referring to is where your genotype is identical, but your phenotype may not be identical, right? And what causes that difference? Some environmental factor, right? And that environment may be your internal environment, like what temperature you had developed at, for example, what nutrition you got as you were developing, or it could be like a much more external feature, such as, I don't know, your social settings or whatever else, right? This could be deterministic. It could be an evolved or adaptive plastic response, such as what you see with these helmets in Daphnia. This is a crustacean that lives in water and forms these really spiky helmets when it senses predation around. So these are larvae of the Daphnia that as they're developing, if they have predator cues in the water, they will have these long spiky helmets developed. But Daphnia that develop in waters without predators will not develop this costly armature. Yeah. But they have the same genotype. Yeah. Right. So this is what is the adaptive plastic response in this case, right? Where it's responding to environmental cues to determine one kind of phenotype versus the other. So for example, imagine a set of genes are turned on only under certain conditions, right? But otherwise they're turned off. I'm simplifying things, but as an example, right? So that your genotype doesn't change, but encoded in the genotype is the ability to change expression levels, modulate them, and so on, right? So some genotypes, yeah, so some genes may regulate the expression of plasticity. Yes, is that what you were asking? Okay. You may also have plasticity that is an outcome of stochasticity, right? So for example, there are errors that occur during transcription. So when you're going from DNA that's copied into mRNA or from mRNA to protein translation. So the machinery that does all of this in our cells is prone to errors, makes mistakes. And those mistakes can then lead to differences in the outcome, right? Why? Because you're producing less protein or more protein, you're producing slightly different protein because you're introducing different kinds of amino acid sequences at some small rate, right? And so that is again, that's going to look as plasticity, but it's not really sort of dependent on environment cues necessarily, and it's not responding to that, it's just stochastic variation, right? So it's kind of noise. I'll just quickly end with one example from work that we did some years ago in our lab. This was led by Lasya, a postdoc. So she had these cells of E. coli, which were more prone to mistranslation. Okay, so these are cells that had a higher rate of translation errors, and this is because she had deleted some important tRNA genes in these cells. So they ended up starting translation at somewhere else in the gene, okay, instead of the start site. So what we found then is we measured how much error was being made. Actually, she measured this in her PhD work, and she found that more errors were being made at the protein level by these strains. We measured the amount of phenotypic variability that was found in these cells in terms of how big the cells got, how quickly they divided at the single cell level, okay, in microfluidic devices. And we found that these guys had lower phenotypic variability, and the mistranslating cells produced more phenotypic variability. This is a cartoon, this is not data, right? And what we found then is that mistranslation is deleterious in optimal environments. So if you grow them in nice, rich media, these strains don't grow as fast. Okay, they make more mistakes, they produce less functional protein, they start translation at random places and so on. Not entirely random, but not at the right place. But if you have a stressful environment, such as some antibiotics, the mistranslating cells actually perform better. And we think part of this is because some heterogeneity in key parameters that allow cells to survive in these antibiotic stresses, the mistranslating cell is more likely to generate some of those cells which are poised to be able to survive and deal with antibiotics, right? So this is not an evolved plastic response kind of situation. It's stochasticity, it's error, but there's a situation where that error can actually be beneficial, right? Is that why this error rate has evolved? I don't know, and we're not commenting on that, right? What we're saying is, depending on the environment, this error or noise can sometimes be beneficial, okay? And the variability is key to that. All right, I think that's the end of it. I'm gonna stop this. Okay. Any questions before lunch? I know everybody wants lunch. Excellent. Lunchtime. See you.